This is my title, The Hidden Half, and I'm going to take you through a wee journey through plant, microbe and soil interactions. The scope of my talk is going to be thus. I have two introductions for you. One is to the Arbuscular Mycorrhizal fungi, and the second is to how we work with them as a community ecologists using DNA. They're somewhat resistant, as you'll see, to any kind of traditional measurements or sort of field assessment. And to illustrate that, I'm going to give you three case studies that are kind of roughly chronological. Bluebells, my absolutely pet plant. Hetchel Wood, which is a fascinating site, which I may talk about too long. And slightly more prosaically, but quite interestingly, the Leeds University farm, where the wood meets the field. So we're going to talk about space and time and communities, and I'll give you some reflections on that at the end. The Arbuscular Mycorrhizal fungi, I first encountered these basically when I was working at the Natural History Museum. We were in the process of trying to relocate out of London, and I sort of applied for a random postdoc that came up in roughly the right area of the world. It happened to be in York, and it happened to be with Alastair Fitter and Peter Young, who were looking at, had a project to try and develop molecular methods for looking at the Arbuscular mycorrhizas. My biggest challenge at the time, I cannot lie, is spelling mycorrhizas properly. So in my application, that was in the mid-90s, mid to late 90s, and it's come a long way since then. So this is the sort of brief introduction to this enigmatic and highly influential group, but they are a bit mercurial. They're difficult to get, literally and figuratively, to get a hold of. So they are root endophytes. We've got the image here, shows these are the external spores, which kind of hang around in the soil on the external hyphae. We've got a diagram here of what they do. They're endophytes because they live inside the plants, in the plant roots. So they're not like ectomycorrhizas, which cover the root. And you can see here, they attach to the surface of the root, and the hyphae weave their way between the cells until they get to the cells that they want to fully colonize with the arbuscules, which I believe is Latin, possibly Greek for little tree, because that's what they look like. And you can see here a micrograph of kind of what that looks like under an acid fuchsid stain. That's not the easiest way to deal with these things, particularly when you're not good at microscopes and you have an astigmatism, as I do. So microscopy, not my favorite thing. They're also very ancient, considered to be have co-evolved and had to have been part of the process of plants colonizing the land. Just to give you an example of that, so here's a lovely micrograph from James Merriweather, who may be well known to some of the people in this room, if not all. It's a photo from his work at York of an arbuscule in a bluebell root, and that photograph dates from about 1995. Here's another micrograph of an arbuscule in a root, and that's from a 400 million year old fossil. They haven't changed very much. They don't have an awful lot of morphology, and that's one of the problems that we have with this particular group, is that they lack the kind of morphology traits that traditional taxonomy kind of demands. So we've had to do look at this. This is from the Ecology, a paper on them. It might be from the Rhine chart, but I'm not going to stake my reputation on that. I'd have to look up the paper again. So they've been around a very long time. Because of that sort of co-evolution thing, they're also ubiquitous. They're thought to colonize approximately 2/3 of plant species and 80 to 90%, depends which paper you read, of plant families. They have a global distribution. They're present on every continent on the globe, even the offshore islands of Antarctica, where they've been found in some of the grasses there. So they truly are a global taxonomic group. They are obligate biotrophs, and they're biotrophs because they gain all their carbon from living plants. They have no saprotrophic ability at all. They cannot decompose dead matter. which many of the fungal groups do, and they're obligate because that is their only source of carbon. So they cannot they cannot generate it from dead material in the environment, and they cannot passively take it up externally. There's no evidence that they can pick up carbon through their external mycelium. They have to get it from the internal mycelium within the roots. It was long thought that that source of carbon was hexose sugars, and I think that's definitely a thing, but it was relatively recently discovered that they also take up fatty acids out of the plants. So there's quite a complex interaction there. And so they gain carbon from the plant, and that exchange for a phosphate is a key nutrient, but also nitrogen. And these are widely considered to be the primary exchange that drives this particular mutualism. But they're a complex set of organisms because they occupy this very complicated niche. They don't sort of just in the environment find something that they decompose and then grow in it and then sporulate and move on. That's not how they work. They occupy two completely different physical niches, both the root and the soil. These things have different characteristics, and the fungi themselves do different things. They take up nutrients in the soil and they take up carbon inside the root. So the root niche is primarily for carbon acquisition, and the soil niche is primarily this nutrient uptake thing. And then so they're continually shuttling things from the outside to the inside, but they're also shuttling it between the plants that they colonize. So they don't colonize one plant at a time. Each plant will be colonized by several different strains, and each strain will colonize several different plants. And that's really interesting in an evolutionary context because it means that the complexity of the interaction means that coevolution is very, very difficult to generate because it's too ephemeral. There's not enough predictability in who collaborates with whom, as it were, for tightly co-evolved interactions to evolve. So where that leaves us is that we have an environment and climate will directly affect the soil niche and how the soil mycelium behaves, but it will indirectly affect the root niche by how it affects the plant that donates the carbon. So that's a complex interplay. And therefore, much of the biology of the fungus itself will be determined by both soil and climate and the interaction between the two. However, the interesting thing about these plants, the symbiosis, is that all of our knowledge about what the fungi are doing mostly comes from the root niche, which I will come to later. And all of this is incredibly difficult to observe directly. Because they're difficult, they're very, why I chose these organisms to work on, I don't know. It would have been much simpler to have something simpler. They're not tractable fungi for traditional mycology because you cannot grow them in axenic culture. This is what a mycorrhizal culture looks like. And it's a plant in a pot with an isolate growing in the roots of it. And if you want to look at its spores, quite often the strategy for getting at spore morphology for taxonomy purposes is to grow these cultures and then you dry them down because that promotes sporulation in the culture because that's their survival organ. And then you extract the spores and you find out which one it is. It's a bit laborious. And it's very difficult to measure function when you've got this other plant and the soil in the way. So this is a typical fungal culture. The closest you can get to sterile culture, sterile axenic culture in arbuscular mycorrhizas is transformed root cultures. So there you have to have a plant that is tractable to become a transformed root culture, and you have to have a fungus that will colonize transformed roots because not very many of them will. So it tends to be the kind of lab rat. There's a suite of lab rat fungi that you can work on in these kinds of systems. And so where that leaves us, you see, and going back to the coevolution thing, is that research on the arbuscular mycorrhizal fungi particularly has actually emerged from plant science rather than mycology, because it became quite clear to a group of plant ecologists, and there's a famous quote about this somewhere, which says that when you're looking at the ecology of a plant, you aren't looking at the ecology of a plant, you're looking at the ecology of a symbiosis with mycorrhizas, because all plants have them in the wild. And so it's really quite interesting how the net effect of this has been is that we as a discipline, we tend to take a slightly plant-centric view of this particular symbiosis, which it's interesting. It's hard to get folk to think about the biology of the fungus and the fungus as an independent entity, because it doesn't have any independence in nature. So it's difficult to maintain what we call a mycocentric approach. It tends to be phytocentric. And it's quite interesting reading papers about the symbiosis and filtering it through that lens of, we're talking about what it does for the plant all the time. And it's quite difficult to push people into thinking, well, what's in this for the fungus? What's the fungal biology here? What are they doing? And so there's quite a lot of debate about mycocentric versus phytocentric approaches. So they're not easy to work on. So how do we do this? And the real game changer in the early 90s came with DNA technologies, when PCR and increasingly high throughput sequencing, this has massively progressed over the last 20 years. We can use molecular ecology methods to study the ecology of all cryptic organisms, but particularly these. For the first time, using targeted methods, you could pull out from field samples which fungi were colonizing a given root system independently of the plant. So prior to that, if you had a multiple colonization, it was very difficult to get microscopic data that you could get total colonization and you could get some morphology of the difference, but there's not enough morphology to detect all of the different isolates in a root. Just from microscopy. That's very challenging. And DNA allowed this to happen. And this technology has resulted in really, really profound changes in our understanding of who lives where, what they do, the phylogenetics and the taxonomy, because we could see how diverse the group was. We can start to look, using genetics, looking at speciation and other evolutionary processes, and the whole community ecology. It's an extraneous of there, I do apologise. To illustrate just how transformative this has been, many of you will have come across Moore's law, which was the prediction, it was an empirical observation that the amount of capacity in computer chips doubled every 2 years. It's actually increasing faster than that now, I think. So it doesn't hold. This is this graph here is a kind of reverse Moore's law, and the cost of sequencing was halving roughly every 2 years until around the mid-2000s. The chemistry of the sequencing technologies changed completely and moved to sequencing by synthesis, and you see this absolute plummeting of the cost of the price per base pair of DNA sequencing, and that's just put the throughput in these systems through the roof. So the challenge used to be getting any data at all. The challenge now is to analyze it and to design experiments which actually utilize that data effectively. The graph at the bottom here is that each of these dots on this plot, this is a logarithmic scale, mark you, is the number of sequences. These are all papers that I'm an author on, and it's the number of sequences in those papers or studies, right? So it's gone from in the late 90s to 84, to the most recent data set that we're analysing at the moment has 5.5 million. And it's just like, it's mind-boggling how much data you get now. So it does often feel a little bit like using a hammer to crack a nut, but anyway, it's great data, and I thoroughly enjoy working with it. Okay, so to parse what we do with the rest of these, with the case studies that I'm going to move on to in a second, is that the way we do this is by using DNA barcodes. This is the botanics, home of much of the methodologies for plant barcoding, and we're basically doing a very similar thing. Mostly we use 18S small subunit ribosomal DNA sequences, and that's our barcode, and that's effectively what we're doing. We're doing community ecology with barcodes. These are environmental sequences, crucially. A vanishingly small number of the sequence types that I'm going to talk about are present or are attached to a named culture that is actually sort of properly vouchered or cultured. There's very, very few of those, largely because many of them aren't easy to culture. Some of them are only really known from spore collections, and it's a complex, the taxonomy of these, validating the taxonomy of these things is a complex business. But they do nevertheless have an identity. When you've worked with them for as long as I have, you meet the same sequence type on regular occasions in particular types of samples. And they start to have, you start to be unsurprised that they are in this sample, but not that one. And they've certainly, for me, it doesn't kind of work for everybody, but for me, there are types that I know have a biogeography in Britain. I know that they're, and we'll see some examples of this, they are more prevalent in some places than others. They're more prevalent in some other habitats than others. So they have an identity. And a game changer in using this sequence identity was the online database Mariam, which was published in 2010, so quite late really, and really only when high throughput data was starting to really ramp up the amount of data out there. It's basically a phylogenetic classification that allows people to use a sequence identity that is comparable across studies. Studies were being published and people were deciding on how to group sequence types in different ways, and this was an attempt to sort of unify that with a kind of phylogenetic and a sensible background. And this is far and away, so post 2010, you will mostly see these papers using virtual taxon or VTs to classify their sequences for the ANs. And yeah, so there are VTs which are really, they're kind of species to me, but I couldn't honestly tell you with any confidence that they're actually identifiably single species, because what is a mycorrhizal species is that's a whole other hour lecture on the genetics of AM fungi, and we're not going there. So that it's complicated, but nevertheless, the VTs have an identity in ecology. Okay, so onto our case with studies. The first is the early years of my work on bluebell woodlands. Bluebells, in this case, Hyacinthioides non-scripta. It's one of the saddest Latin names that I know, a bit like a hyacinth, nondescript, very sad. They have, typically with that group of organisms, they have bulbs, they have limited, very unbranched root systems, and they have very limited foraging ability as roots, okay? They're vernal geophytes, so most of their growth is not during the summer, and so they're a niche that's available to soil microbes during the winter, which is quite unusual. Because of their, frankly, rather rubbish root systems, they're very dependent on Arbuscular Mycorrhizal for peat uptake. The early work on bluebells was done by James Merriweather, who in the field manipulated bluebell bulbs so that they were lacking in mycorrhizal connections, and showed that if you deprive them of phosphate, if you deprive them of mycorrhizas, you deprive them of phosphate and they don't flower. So it massively impacts bulb growth. And when you dig these up in the field, with permission naturally, they're always very heavily colonised by AM fungi. Since I'm back in Scotland, yay, I can ask this question. So I've gone from the odd situation of working on bluebells, not harebells. I now work on English bluebells, not bluebells. But there you are. I also have to put that joke in. So their field site was the it's a very glamorous field site, Pretty Wood, in Castle Howard Estate. We weren't very near the house. We were, however, near Four Faces, which is a folly in the middle of Pretty Wood, and that was the kind of quadrat pole for our sampling sites there. And so we were following up the original studies done by James Merriweather, incorrectly spelt there, and Alastair, when I first moved to York. Right, so this was the very first attempt to classify and quantify mycorrhizal communities in a field system using DNA, or arbuscular mycorrhizal communities, I should be specific. What we did was we chose several sites within the woodland under different canopy types. So we figured that the majority of the root community available to the fungi, bluebells aren't the only roots in these systems because it's a woodland, and the major root systems are from the trees themselves. So we had sycamore site dominated sites. Sycamore is an arbuscular mycorrhizal tree, and we had oak woodlands on the site, which is an ectomycorrhizal tree. So we would predict that they would be quite different. But we also sampled at 2 different times of the year. At the end of the season, the root season in July, they kind of senesce after that, and they're more or less rootless during the summer, and then the roots begin to grow again in September, and then we resampled in December. The AMF were identified using a cloning technique. It was incredibly laborious and it was incredibly expensive, and I had the grand total of 84 sequences for this study, and that was loads at the time. Anyway, and this is what we came up with. So we can see here we've got 4 stacked bar charts, relative abundance of these different types of fungi, and you can see that in the July, the 2 canopies are distinctly different. There is a lot less of this bright green one under the sycamore canopy than there is under the oak. They're Ecology for the taxonomy nerds. But the same is true in the winter. The canopy types still differ in different ways, but the dominant taxon has completely changed. And what you see in the down season is this Scutelospora type, which James was very aware of from the spores. We knew it was there, and we were unsurprised to find it in the roots in the winter. What's really interesting about this particular taxon was that it was clearly kind of winter adapted. We speculated that it was either cold tolerant and not very competitive, so it was able to proliferate during a time when other taxa weren't there. And we had some circumstantial evidence in favour of that, in that it was absolutely impossible to pull it into culture. And I suspect we would have had more joy getting this one into culture if we'd actually cultivated it in the fridge, because I suspect that was its favoured habitat. Okay, so later we did a lot of work in Prettywood, and we got quite a good idea of the fungal taxa that are there, and that's just one bluebell woodland. How true is that more widely? And what I really like about a British Isles-wide survey of bluebells is that you're effectively studying the entire global distribution of a species, because most of them are in the British Isles. And so what we wanted to look at was, well, okay, we know about Prettywood, which is an oak woodland with bluebells in it, and the 2 dominant taxa in these woodlands are effectively oaks and bluebells. What are other oak and bluebell woodland sites like? And because they tend to be quite plant species poor, you can kind of ask that question by looking at other oak bluebell woodlands. And so we identified oak-dominated bluebells, bluebell populations in 6 regions of the UK, 3 up the east coast and 3 up the west coast, and with paired sites, with 2 sites in each region. All dominated by oak and all relatively species poor undergrowth, except for bluebells. Using this, you can then ask the question, what are the fungi tracking? Are they tracking climate? Because we know that there's a significant climate variation across that latitude and that longitude. Or are they tracking the host? Because if they're really tracking the host, then they're actually host specialists, which was one of the questions we were really trying to get at. There was a lot of question whether these relationships, once we discovered the diversity of fungi out there, how evolved are they as host fungus pairs? And there was some indication that there's preference, but this was a really good field experimental system to actually test that and say, if they really are specific to bluebells, then they will track the bluebells wherever the bluebells are. If they're not, they won't. This is what we found. Oh, I should also say, in this case, we're now into next generation sequencing, and we're at 50,000 sequences. So this is a little bit more intense. Right. Okay. No, that way. So what we've got here is the map again, and we've got they're in alphabetical order, which makes the parsing of it a little bit tricky. But we've got the western ones on this side, and we've got the eastern ones on that side, and you can eyeball immediately the difference between the east and the west. There's quite clearly a set of dominant taxa that are predominantly eastern, and the western ones seem to be a little bit more diverse. The grey bit at the top, these are the top 15 taxa in that have colours, and that's 90% of the total amount of fungus that we counted. The rest of the diversity, which is, you know, another 25 different taxa roughly, fall into this grey category, but they're so those patterns aren't so clear. So we took the top 15. What was really interesting here was that what we found in the Yorkshire Prettywood site were also typical, but only in Perth and Sussex. And there are some taxa which were really dominant in Prettywood, which only also occurred in the Perthshire sites. So this was a sort of northeastern, there was clearly a northeastern type, there was clearly a western suite that were in common. And what you can also see here is that this brown one is northern, and because it's present in both Highland and Perth and in very low numbers elsewhere. Okay, so we can start to see this kind of biogeography emerging. And just to really visualize that more clearly, we plotted this ordination, and this is the first of several of these ordination graphs that I'm going to show you. Basically, each dot is a sample, and the closer the dots are together, the closer the points are together, the more similar their communities are. So it's a way of clustering and seeing where the clusters, which samples cluster together. In this case, this is a fitted ordination, and it's fitted to Met Office 1 kilometre square climate data for the Ordnance Survey grids that these samples were taken from. And you can see very clearly that rainfall is associated with the western ones, funnily enough. Continentality, which is the difference between the July maximum and the January, actually the February minimum, because that is the coldest month apparently. The higher that number, the higher the continentality, so fluctuation between summer and winter. Continentality drives the eastern ones. I think that was really interesting. And the number of frost days is kind of driving northern and eastern. So that starts to give us a steer that at this scale, we can very clearly fit a sort of biome type classification onto what kind of communities we would expect to see. There is a seasonal dynamic still there. You do see a change between the two sampling times, and there is a lot of between site variability. The variances are quite big, so I can't gloss over that. But nevertheless, there is some variation influencing this, but the within site differences that we saw in Prettywood on its own still operate in each of these sites. But when you put them all together, you do see this regional pattern emerge. And we can start to sort of speculate that actually there's something around maritime versus continental climates here. That's not going to be a direct influence on the fungi, but it must be an influence on how that affects the soils, how that affects temperature and things like that. And we'll come on to that. Okay, right. Second example is Hetchelwood. This is a great site. I'm going to guess that nobody here's ever been there. a possible exception. Hetchelwood is this is a Google map of the site. It's kind of between the M1 and the outskirts of Leeds. It's a Yorkshire Wildlife Trust site and an SSSI, and it's just the oddest, one of the oddest places I've ever been to. Its geology is an SSSI both for its geology and archaeology and for its calcareous grasslands. So it's botanically, the calcareous meadows are very, very rich. They are, the meadows up here are limestone-rich grasslands with some really interesting species in them. There is an escarpment here and that goes right round, and the land drops quite dramatically. And what you get here in the Pompacali earthworks is lowland heath. So within spitting distance, you have species-rich calcareous grassland and lowland heath. And the reason for that is that you've got limestone sitting over acid millstone grit. And so wherever the limestone's been eroded away, you get a very, very sharp change in soils and pH. And the reason why you get this area of lowland heath is because it's a Roman quarry, and they quarried all the limestone away, left the millstone grit, spoil heaps behind, and lowland heath has developed on it. It's a fascinating site. So you get this really weird sort of earthworks landscape in this hollow, and then all this limestone escarpment around you. Absolutely bizarre. So again, we were wanting to test this. What do the fungi track? Do they track their host or do they track the environment? We had an indication that there was a sort of climate effect. The soil pH is a much, much more direct driver. And that's what we wanted to look at. So on the escarpment, we work on the escarpment where the limestone is eroding away, and then you get this limestone grit. That's this transition here, which you can see on the map. And that's a 3D plot of the pH on the site. It's just, so it's 20 meters, it's not quite to scale. This is 20 meters. These numbers are centimeters, right? This 20 meters translate to long, and then a pH gradient in front of you. The y-axis here is 7 meters. So if I'm standing here at a pH of 3.4, the door out of the lecture theatre is at pH 8.2. It's just, and you can just see it. You can see it on the ground. The vegetation changes just in a space of about 2.5 meters. It's quite extraordinary. But some plant taxa can cross that gradient, others don't. So you've got a really, really nice way of testing how the symbiotic fungi track the host plants given this environmental gradient. Because if they're absolutely tracking pH and not hosts, then that's where you'll find them. If they're tracking hosts, then they may cross the gradient and you won't see, you know, so you can start to sort of, all of the potential permutations are there. So this is an aerial view of that same site with the crosses where we got the samples from. We're into 425 root samples. This was not done using sequencing. This was done using a different method. A fingerprinting method was used for this. And but it was a lot of individual root samples. So individual root samples were taken so that we could also sequence what host it was coming from. So you knew what the root was, and you knew the fingerprint of the community that was colonizing it. We've got another ordination here, and we've divided this up in symbols to 3 groups. We've got a low pH group in black, which is like pH 4 and below, the sort of medium pH of around averaging around 5.5, and then anything 6.5 and beyond is in the open squares. And you can see very clearly that the fungal communities are not tracking the host primarily, they're tracking the pH. So there were very many confounders in this data set, largely because although there is a linear gradient. So the gradient of pH only goes in one direction. So that means you get, you have to do quite a lot of analysis to make sure that you're not putting spatial autocorrelation in, because they'll be more similar by chance because they're nearer. Nevertheless, once you account for that, there is still some plant host species preference in there, but it is not as strong as the response to pH. So variation in space because of environmental factors at the regional scale and at the local scale is clearly important for fungi. They are tracking and surviving in optimum soil conditions for them, and they're more likely to be abundant in soils that suit them than following their host around, because the presence of the host is probably too unpredictable for that to be a good strategy. Okay, we did another study at Hetchelwood, which was a time study. In this case, we took a just checking which it was. Yep. Okay. So this time, here's Hetchelwood again. Here we've moved east into the limestone grassland itself, and we took 6 samples from roughly the same meter square, 10 times over about 10 months, 8 to 10 months. So this was quite a deep time series, and which is unusual. It's very unusual. This is not easy data to collect, and therefore people do tend to take one set of samples, and there's not very many time series in the literature. Here we've got pooled root samples, 11 time points in 8 months, back to high throughput sequencing, 108,000 sequences this time. And here again, the ordination, very, very clear differentiation into summer samples and winter samples, right? So both time and space are important. And what was really, really striking about this particular set of data was how quick the transition was. And this is a slightly horrible table, and really should make a better graphic for this. But if you note the date up there, 7th of April and the 17th of April, right? And this is the number of reads that you get for a given, each of these is a type of sequence, right? And so you get all this set of taxa completely disappear in 10 days. It's the same meter square, right? And they're just gone. This is possibly a relative abundance problem. It's probably because these are declining in relative abundance because this lot have appeared out of nowhere, right? These are probably taxa that survive as spores that germinated and colonized between the 7th of April and the 17th of April that year. We did some modeling on this, and the highest correlation we got was to soil temperature 2.5 weeks prior to sampling date. That's very likely correlated to there's a flush of root growth and something happens. But the speed of that transition really surprised us. It's very much more dynamic than people thought. Final case study then is where the wood meets the field. So we're increasingly thinking about regenerative agriculture and about how we use soil ecology and soil biodiversity to maximize and improve food security. And we were involved in a big consortium looking at how lays, fallows, sort of long-term fallow in agricultural systems changes soil microbiology. And it's a comparison between this rhizosphere development between lays and conventional arable, but also between the hedgerows and the field margins of that site. So this is what this experiment looked like. It's the most preposterous experiment, a field experiment I've ever been involved in. This is basically it's a plough width, 70-meter long strip that was put into arable fields, connected to the hedgerow, so that you could see if there was any migration between them, and managed as a ley strip within the arable field itself. So you had a direct comparison, and very, very big. Only possible because Leeds University owned the farm and could tell the farm managers exactly what to do. So this would not be an easy experiment to do otherwise. So you can see here, this is the ley strip. It was mowed periodically. This is the arable, and this is the hedgerow that it connects to. So we've got these different land uses in there. Okay, so that's what it is. There are long-term land uses here, arable, the grassy margin, and the hedgerows themselves. They were assessed as being sort of average condition UK hedgerows. And this is what it looked like on Google Maps. If you go on Google Maps now, it's all gone. You have to go back to the old data to see it. But you could pretty much see it from space. These are the ley strips that you can see. And this tells you roughly what, there we are, that's the grassy field margins and the hedgerows. The short-term land use change was these ley strips. Okay, right. So one of the questions we were very interested in is, if you stop disturbing the soil and you connect it to a refugia, so effectively, hedgerows are really important linear features in landscapes that act as refuges for animals, plants, and potentially soil microbes. Would they move and colonize this this now undisturbed extension to the grassy margin? And in this case, what I'm going to show you is data taken from 272 root samples taken over 5 time points between 2016 and 17. And now we're up to 1.5 million sequences. So we're starting to see the throughput increase. Final set of ordinations. Here we have all of those samples put into the graph. The shape is the different fields, because the field is the replicate essentially, and the samples within the field are pseudo-replicates. But we have M is the margin, H is the hedgerow. They're all coloured in a green colour, and then the numbers are the distance from the margin. There's very little effect of space within the arable field at all, so you can kind of ignore that. Basically, the important thing here is the green and the brown. The hedgerows and the grassy margin are totally different from the arable and the ley. So the short-term land use change hasn't made as much difference as we expected. So they are different. How are they different, I hear you ask? What you can also plot on these ordinations is the taxa, the members of the community in each of those dots. You can see which ones are important either side of that graph. And that's what you can see in the red. You can see that they're VT numbers, in fact. So there's some old friends there. If you look really carefully, oh, that's too small to see. So all of the in the arable fields, so what we can then do is, going back into the Marianne database, we can see where in other studies these taxa have been found. Funnily enough, there's a whole batch of them in the arable site that have been found in arable crops elsewhere. And indeed, I saw a piece of work about a European survey which had found all of our taxa in their fields as well. So this is not unsurprising. In the grassy margins, one of the ones is from a typical grassland. As you get further away, we find the woodland types. We find types that have been found in woodlands, and a whole bunch of them that we found in pretty wood and bluebells. So, and there are bluebells in those hedgerows. So it all starts to join up and make sense, which pleased me greatly. So what we're seeing here is, and for some strange reason, we have acidic upland grasslands in there. Possibly that's not necessarily that surprising, but these are all types which kind of make sense. You've got the, on this side of the graph, you've got the acidic upland grassland types. On this side of the graph, you've got the arable types, okay, which is entirely consistent with our understanding of where they are. So what we get here is that the hetero AM communities are really distinct from the other communities, even though they're right next to each other. Right. So again, here's a space. This is a long-term land use which has created diversity within a small amount of space. And it's likely driven by the host assemblage, and particularly through lack of disturbance, which is a significant factor in determining what happens in arable fields. There is some differentiation between the ley and the arable, but it's a change in relative abundance. It's not a change in composition at all. There's absolutely no evidence over the time period of this study that the taxa from the hedgerow migrated into the ley, none at all, or vice versa. So there's the dispersal limitation. There is something limiting the dispersal of these taxa. Okay, so this is a sort of slight sidetrack, and since we're in very close to a very fine herbarium, I have worked in a herbarium, and literally where my desk was in the herbarium. So I came into this kind of molecular ecology with that kind of curation head on, and I've never thrown any of these samples away. So this is the cupboard in York where my soil and plant material archive was stored. It is now all under one of the lecture theatres in Ashworth. I brought it all with me. I wasn't going to throw it away, because these are the only voucher specimens we have for these taxa. Actually, is samples like this. So, and the beauty of being able to keep them and curate them means we can go back to them. So if we get better techniques to actually look at these things, I can go back to these samples and do this again if somebody will give me the money. So I do worry about these things. So. Quick sort of summary. The AM community, the fungal community, depends on the plants around them. We know that. Not just the plants that they colonise, but the other plants that they're not colonising also have an influence. So the community depends on where it is, independently of the host plant. So all of these environmental factors influence the biology of the fungi and where they occur. What's interesting is that soil characters, and notably pH, is also a significant driver of community composition. This is becoming an axiom in soil microbial ecology, is that the first thing you look at is pH. It is a kind of master driver of many things, and many organism groups in soils are primarily defined by pH. And we probably need to stop looking at pH and actually look at what pH is a proxy for. But it's interesting that the AM fungi are doing that as well as other microbial groups. They're incredibly dynamic, much more dynamic than we thought, and they show distinct changes in time as well as in space. So they can be diverse across short distances, but they're also diverse in short time periods. And that's an area that's massively lacking in the literature, is timing. From a sort of conservation biodiversity point of view, landscape diversity encourages high diversity overall. And I think that's a really important message. We know that's true for macrofauna. It is true for microbes and microfauna flora as well. And a really interesting emergent idea from our studies is that some groups of fungi, particularly, not true for all, may significantly lack the ability to disperse, and their sort of geographic limitation may actually be dispersal limitation rather than environmental adaptation. And if that is true, then their resilience to environment change may be quite low, and that's a conservation thing for us to think about. So to pull that together, I initially started working on AM fungi to look for host specificity, for evidence of host selection and host specificity, and how plants would select fungi from background, very phytocentric view. But it's turned out to be much, much more complicated than that. As we can see from just these case studies, and there's a whole lot more that I could have talked about, their distribution and diversity is likely to be a function of the fungal traits, the actual biology of the actual fungi. And that I won't say the host plants are bit parts, but they're only one part of the conversation. And unfortunately, those traits and that biology is the most difficult thing to measure. But despite, I want you to come away thinking, because we talk a lot about symbionts, and this particular group of symbionts are very, very connected. They're very connected to their hosts, and they're connected to each other, and they connect hosts together too. But they have a biology and they have a life of their own. And it's my plea to you when people talk about symbionts, not to forget that the symbionts have their own lives too. Long list of acknowledgements there for the 3 projects I was involved in, with credit to the funders as per. And I will leave you with this quote from Jacob Bronowski and invite any questions. Thank you very much.